Hello and welcome to the urinary system pre-lecture. Just to make sure, you should have your learner response sheet in front of you, and you can really just fill it out as we go through the presentation or just shortly after. You'll just point form, really basic questions 1 through 4 you'll be filling out during the presentation. So the purpose, which again relates to your sheet that you're filling out, is to just serve as an introduction, get you oriented and somewhat familiar with the urinary system before we cover it in more detail in lecture. So we'll briefly go over the anatomy and then also go into the process of urine formation. Again, just make sure you do have your sheet with you. If for some reason you've misplaced it, you've lost it, I did post it on WebCT. You'll need to print it, and you really just need to print page 3 because that's actually the sheet that you're filling out. So if we look at the anatomy of the urinary system, there are 2 kidneys, 2 ureters, a urinary bladder, and a urethra. And just a note here that the urinary system is closely related with the circulatory system, and it's the location where blood is literally filtered of any waste or any items that are in excess that the body doesn't want, the body doesn't need, and that forms the urine, and then we get rid of that through excretion. So one of your lecture objectives here is to be familiar with and be able to label the structures of the urinary system on a diagram. So I've provided this diagram with you here that you can use to memorize. Essentially, we have the kidneys are found in the abdominal cavity, just the posterior abdominal wall actually. So there are 2 kidneys, and then the ureter is the tube that passes from the kidney down and basically carries the urine to the urinary bladder for storage. And then another tube is called the urethra, which is basically the tube that causes the urine to exit the body to the outside. So, if you were to picture that we were slice this kidney sort of in half, you would picture about 1 million to 2 million nephrons per kidney. So the nephron is really the microscopic structure of the kidney where urine formation actually occurs. Blood filtration leading to urine formation is occurring there. So, 1 to 2 million per kidney, and then essentially as the urine gets formed, it really just drains into the center area of the kidney, gets collected there, and then travels down the ureter, which is really just a passageway to the bladder where it's stored, and then the urethra where the urine exits. So a little more detail is what we want to go into here is the process of urine formation. So there are 3 main steps: glomerular filtration, tubular reabsorption, and tubular secretion. And again, just relating this to the lecture objective, you'll need to define and understand the process of glomerular filtration as well as tubular reabsorption and tubular secretion. So here are the definitions. Essentially, we'll just focus on glomerular filtration, and what happens here is small molecules leave the blood and they enter the nephron, essentially the part of the nephron, the tubule. So we'll just look at this diagram here. Just to orient you, the yellow structure here, there's sort of like this little capsule, the circular capsule, and then this series of coiled up tubes here. This is all known as the nephron, and this first section is like a little cup that catches the recently filtered blood. Once the blood is filtered, that substance is called the filtrate, and that enters into the first part of the nephron called Bowman's capsule, or sometimes referred to as the glomerular capsule, and it catches. So again, imagine there's like 1 to 2 million of these nephrons found within each kidney, and they're each sort of filtering out and fine-tuning or processing the filtrate to create urine. And you can see it's closely related with the blood. So we have the renal artery, which branches off, and each branching forms an arteriole, and each arteriole supplies 1 nephron. So there's, again, 1 to 2 millions of these. So at glomerular filtration, there's tiny little pores within these capillaries. So this tuft of capillaries is actually called the glomerulus. And so that's where the term glomerular filtration came from. And the pressure of the blood passing through here literally pushes substances out of these tiny little pores. But they're a certain size, so larger substances like cells or large proteins actually cannot pass through the tiny little pore in the membrane. So things like water, amino acids, glucose, ions, and of course waste pass through and enter into Bowman's capsule. So once that's sort of filtered out of the blood, now that liquidy substance containing all these items is called filtrate. So if I bring us back to the slide before here, things like water, glucose, nutrients, waste, salts do pass through, and that's known as the filtrate once it enters. But again, things like cells, which are quite large, and larger macromolecules like proteins, they just cannot pass through. So normally in somebody's urine, you're not going to find red blood cells. You're not going to find proteins. That's an abnormal finding. The second stage of urine formation is tubular reabsorption, and that really occurs in the first part of the nephron. So essentially a lot of the molecules get reabsorbed, and reabsorbed means that it moves from the filtrate in the nephron back into the blood capillaries. So we'll just go to the next step here, tubular reabsorption, and you see these items that entered into this filtrate. So now imagine they're in this yellow coiled tube, right? They're draining down here, and it just continues. And essentially at this point, you see reabsorption occurring. So the glucose that's in the filtrate goes back into the blood, and some ions that were filtered out move back where they came from in the blood, hence the term reabsorption. And same with water. You know, you filter about 180 liters of water per day. You only urinate out about 1 to 2 liters. So where does the rest go? Essentially, it's not lost through urine. It's all reabsorbed. So again, glucose definitely gets reabsorbed. Glucose is not normally found in the urine. That would be an abnormal finding. You know, that's typically seen maybe in diabetics who have uncontrolled blood sugar levels. But essentially anything that we need, especially, you know, water we're going to reabsorb, glucose we're going to reabsorb, nutrients, ions, salts we're going to reabsorb. Essentially the body's like, oh, it filtered out, you know, but I want to put it back into the blood. And then the third stage is called tubular secretion. And essentially this is a way of getting rid of substances that maybe weren't removed at the first step of filtration. So often it is a passageway for things like metabolic products from drugs, like penicillin, pesticides. We can actually take that out of the blood and put it into the urine, and the process by which that occurs is called secretion. So you're essentially secreting these substances out of the blood and putting them into the filtrate, into the nephron tubule. So here, tubular secretion. So often it's kind of happening more, it happens all along the tube, but often kind of the last portion of the tube is where a lot of it can occur. And you can see these arrows going into the nephrons. So things like drugs, penicillin, pesticides, some ammonia, some potassium, some hydrogen ions are all entering into the tubule. They're entering into the filtrate. So it's going from the blood into the filtrate. So it's sort of the opposite of reabsorption, where you're going from the filtrate into the blood. And then basically the filtrate now continues into the last portion. Well, it basically joins a structure called a collecting duct, which, as I mentioned before, all these ducts lead into the center of the kidney, called the renal pelvis, where all the blood, or the urine gets collected, drained out, and then proceeds down the ureter. So I did post a video here that I will essentially click on to show you. So if it's not working, you can always just like copy and paste this. Essentially, there's some talk about, you know, the loop of Henle and what's occurring there. You really just need to focus on the processes that occur at Bowman's capsule, at the proximal convoluted tubule, and at the distal convoluted tubule. So in the essence of time, yeah, I think it's about 5 minutes, so we'll just click here and play it. Okay, so just to talk a little bit about abnormalities, and this is what we'll cover also in the lab exercise this week. Sometimes, sorry, I think I'll adjust the volume here. Sometimes due to kidney damage, diseases, or other conditions such as diabetes, substances that should not be found in the urine are actually found in the urine. And this really forms the basis of urine tests and urinalysis, as you can actually just do like a little dipstick in newly collected urine and sort of see what substances might be present and what conditions those relate to and possibly help make a diagnosis. So essentially I've got a few slides here to go over. If you wish to get a head start on the lab exercise, the following slides will walk you through a urinalysis. So essentially, ideally the test is performed within 15 minutes of the sample collection. And so, you know, I'm sure most of you are familiar with this. You have a little urine collection cup. There's your little dipstick called a chem stick. And then essentially you put the test indicator strip into the urine, wait about 2 minutes, remove it, sort of tap it dry, and then essentially you can read. It's based on color, so if the items are present in the urine, certain colors will appear to show presence of certain substances. So essentially, like white blood cells, proteins, it measures the pH of the urine, look at the presence of blood, glucose. So again, things like glucose and proteins should not be found in the urine. And that pretty much sums it up. So hopefully you've been able to fill out your sheet. If not, you can finish it up now and just bring it to